Oxygen enables living organisms to extract large amounts of energy from fuels, from carbohydrates, fats, and, if necessary, amino acids. However, oxygen can accept single electrons to form highly reactive radicals, called reactive oxygen species or ROS. They include the superoxide radical, hydrogen peroxide, the hydroxyl radical, and singlet oxygen. Because reactive oxygen species are so reactive, they can damage living cells if formed in significant amounts. Living organisms employ antioxidants, which are easily oxidized themselves and preferentially react with reactive oxygen species. Under certain conditions of oxidative stress, antioxidant defense mechanisms are overwhelmed, and some cellular damage may occur. Such damage may include enzyme inactivation, polysaccharide depolymerization, DNA breakage, and destruction of membranes. Oxidative damage contributes to the aging process and has been linked to numerous diseases. Several types of cells produce large quantities of reactive oxygen species as a part of their function. Examples are macrophages and neutrophils, which in a process called respiratory burst destroy microorganisms and damaged cells. Oxygen molecule is a diradical. Common representation of oxygen molecule represents it as having a double bond. That would be Lewis structure, and that's valence bond theory. A more accurate description of oxygen molecule is given by molecular orbital or MO theory. And according to its representation, oxygen molecule has two unpaired electrons. We can draw a Lewis formula that would correspond to results of molecular orbital theory, and that's representation on the right. So representation on the right is still valence bond theory, Lewis structure, but that is consistent with results that molecular orbital theory gives us. And that's actually correct representation of oxygen molecule. Oxygen molecule has two unpaired electrons. According to valence bond theory, molecular oxygen should be capable of undergoing both 1 and 2 electron reactions. But only reaction with single electron, which is characteristic of free radicals, is observed. In common reaction, one of the free electrons of oxygen pairs up with an electron to form a superoxide radical anion. In chemistry, it is usually called superoxide anion, and in biochemistry, superoxide radical. Superoxide anion can form stable salts with alkali metals, for example, potassium superoxide, KO2. In cells, it is extremely toxic and is used by phagocytes to kill bacteria. It is also a byproduct of oxidative respiration and must be eliminated to avoid cell damage. In the mitochondrial electron transport, 4 electrons and 4 protons are transported to a molecule of oxygen to give a molecule of water. During this process, several reactive oxygen species are formed, but they are trapped within the cytochrome oxidase until the process has been completed. However, electrons may leak out of the electron transport pathway and react with oxygen to form reactive oxygen species. In addition, exposure to UV or ionizing radiation can result in formation of reactive oxygen species. Cells employ defense mechanisms to minimize ROS damage or reactive oxygen species damage. The first reactive oxygen species that is formed during the reduction of oxygen is the superoxide radical. And it's formed by electrons derived from the Q cycle in complex 3 and by flavoprotein NADH dehydrogenase in complex 1. Superoxide can act as nucleophile, an oxidizing agent, or a reducing agent. As it is lipid-soluble, superoxide radical can cause considerable damage to cell membranes. In an aqueous solution, the superoxide radical undergoes disproportionation to produce hydrogen peroxide, and reaction is pH dependent because it requires protons. hydrogen peroxide itself is not a reactive oxygen species and diffuses out of the cell. In reaction with iron 2 plus ion and other transition metal ions, it decomposes to give hydroxyl radical, and hydroxyl radical is a reactive oxygen species. Hydroxyl radical can cause considerable cell damage because it reacts with most of the biomolecules and, even more importantly, can initiate a radical chain reaction. Another highly destructive species is singlet oxygen. Ordinary oxygen is triplet oxygen. Singlet oxygen is a result of promotion of an electron into a higher energy orbital. So when oxygen molecule absorbs energy, such as UV or energy of chemical reaction. As oxygen molecule is in an excited state, it is highly reactive and is more damaging than the hydroxyl radical. To protect themselves from oxidative stress, living organisms have developed several defense mechanisms, most important of which are antioxidant enzyme systems and antioxidant molecules. Antioxidant enzyme systems that provide defense against the oxidative stress are superoxide dismutase, glutathione peroxidase, and catalase. The superoxide dismutases, or SODs, are a class of enzymes that catalyze formation of hydrogen peroxide and oxygen from the superoxide radical. There are two major forms of superoxide dismutases. In humans, the copper-zinc isoenzyme occurs in the cytoplasm, while the manganese-containing enzyme is present in the mitochondrial matrix. Glutathione peroxidase is a selenium-containing enzyme. It contains a selenocysteine residue. Its role is to reduce hydrogen peroxide and other peroxides such as alkyl hydroperoxides and dialkyl peroxides. For example, in a reduction of alkyl hydroperoxide, the reaction products are the corresponding alcohol and water. Glutathione disulfide is obtained by oxidation of glutathione. The mechanism of reduction of peroxide, for example hydrogen peroxide, involves oxidation of the selenol group of selenocysteine residue by hydrogen peroxide to give the selenic acid, RSEOH, and then the selenic acid is converted back to selenol by a two-step process. It begins with reaction with glutathione to form a bond between glutathione and selenocysteine residue, and a molecule of water is a byproduct. A second glutathione molecule reduces glutathione selenocysteine intermediate back to selenol and releases oxidized glutathione that has disulfide bond as a byproduct. Upon reaction, the enzyme has to be regenerated, and one of the enzymes that catalyzes its reduction back to glutathione is glutathione reductase. And here is a very simplified reaction scheme. The overall process actually is a reduction of hydrogen peroxide by NADPH and a proton to give 2 molecules of water and NADP. Catalase is a heme-containing enzyme that utilizes hydrogen peroxide to oxidize other substrates. Large amounts of catalase are found in peroxisomes, where relatively large amounts of hydrogen peroxide are generated by various oxidation reactions. When hydrogen peroxide is present in large amounts, catalase converts it to water. Another way living organisms protect themselves from oxidative damage is by employing antioxidant molecules. Some of the antioxidant molecules that living organisms employ as a protection from free radical damage are glutathione, alpha-tocopherol, ascorbic acid, and beta-carotene. Alpha-tocopherol is a radical scavenger. It is a phenolic antioxidant. It reacts with free radicals to generate a stable, resonance-stabilized phenolic radical. Since phenolic radicals are stable, they don't cause cell damage. alpha-tocopherol is lipid-soluble and plays an important role in protecting cell membranes. On the other hand, ascorbic acid is a water-soluble molecule, and it operates in the aqueous environment of the cell and extracellular fluids. It undergoes reversible oxidation to dehydroascorbic acid. So ascorbic acid reacts with a free radical, such as hydroxyl radical, to give L-ascorbyl radical, and then in reaction with another free radical, with, for example, another hydroxyl radical, it loses a proton to give dehydroascorbic acid. Dehydroascorbic acid exists in its hydrated form, shown on the right. Besides operating in aqueous environment, ascorbic acid protects membranes through two mechanisms. First, it reacts with peroxy radicals in the cytoplasm before they can reach membranes. Second, it enhances antioxidant activity of alpha-tocopherol by regenerating it from alpha-tocopheryl radical. Ascorbate that is produced in the process is then regenerated in reaction with glutathione. Beta-carotene is a plant pigment from the class of carotenoids. In plants, they play dual role. They absorb light necessary for photosynthesis and protect from reactive oxidative species damage. In animals, beta-carotene is a precursor of retinol, or vitamin A, and an important antioxidant in membranes. Retinol is precursor of retinal, the light-absorbing pigment in the rod cells of the retina. And this completes our lecture on oxidative stress.